All right, so we are going to talk about biomimesis and scientific method. So, what is biomimesis? Biomimesis essentially is the imitations of natural processes to advance human objectives. The human objectives could be to develop new technologies, to formulate new medicines, or have new innovations. So, biomimesis is just you imitating natural processes to advance your objectives. And your objective could range from you developing a technology, medicines, and other innovations. While scientific methods is actually a systematic approach to acquiring knowledge and understanding of the natural world. So first, you have to understand, you have to acquire the knowledge and understand the natural world before you can imitate it. So it is very important. Scientific methods will actually equip you with that empirical evidence and logical reasoning, you know, that will aim to develop accurate and reliable explanations of the natural phenomenon, right? So you see, biomimesis, then scientific method. You have to first understand something before you imitate it. You understanding something is about you getting the knowledge for you to have, you know, um, an empirical evidence and logical reasoning to explain certain things. And then I talked about evidence. What is an evidence? Evidence is an absolute term, meaning an unambiguous, reproducible prediction. Evidence is an absolute term, meaning an unambiguous, reproducible prediction. There is no evolving evidence. If there is no evidence, there is no evidence. There is nothing like evolving evidence. That's what makes it absolute, right? Now let's jump into the some of the key steps in scientific method. Scientific method starts by you observing and describing a phenomenon in the natural world. You first have to observe something and then you describe it. And thereafter, you formulate a hypothesis that explains the observed phenomenon, right? Now, after formulating a hypothesis, you have to design and conduct experiment to test the hypothesis, right? Now, the next thing is to analyze the data collected from the experiment to see whether they are in support of your hypothesis or not. And of course, you have to draw your conclusion and refine the hypothesis on the result obtained. And finally, you have to communicate the result through publications or presentation for people to read, to see gaps they can, fill in for further, research work. So that's essentially what scientific method is about, right? Now, I want to talk to you about research approaches. Research approaches differ from research designs. When you talk about research approaches, there can be quantitative, there can be qualitative, and there can be mixed method research design. Under quantitative design, we have the descriptive research design. Now, this descriptive research design aims to observe and describe characteristics of a population or phenomenon without manipulating the environment of variables, right? There is no manipulations of variables in descriptive research design. And most of the work on prevalence, prevalence studies, essentially, they are basically descriptive or what we call the observational research design. Then we talk about the correlational design. It examines the relationship or association between two or more variables without manipulating them. Again, there is no manipulations of variables in correlational design. All you need to do is to examine the relationship or association between two or more variables, right? That's the way it is. So you can talk about exploring the relationship between physical activity level and then the blood pressure. Now, the third one is what we call the causal comparative or the ex post factor design. This one compares two or more groups that differ in one key variable to determine possible cause and effect relationships, though the cause is not manipulated. There is no manipulations of variable here too. Example, comparing the academic performance of students from public and private schools to infer influencing factors, right? Now, this is where the gist is the experimental design. When you talk about experimental design, it encompasses both dependent variable and independent variable. Independent variable is the variable that is manipulated or controlled in an experiment. The dependent variable is never manipulated or controlled. So in experimental design, you have both the dependent variable and the independent variable. So it involves essentially manipulating one variable, which is the independent, to observe its effects on another, which is the dependent variable, under controlled condition. Example, testing the effect of a new herbal extract on bacterial growth in laboratory settings, right? Now, let's jump right into the second arm of the research approaches, which is the qualitative research design. Remember, I told you under this, we have what we call the phenomenology design. This one focuses on exploring and describing lived experiences of individuals regarding a particular phenomenon. Example, understanding patients' experiences living with chronic hepatitis B, right? Now, the second design under the qualitative is what we call the case study design. Case study design provides an in-depth exploration of a single case or a few cases within a real-life context. Example, analyzing the management of an outbreak in one hospital as a case study. The third one, we call it the grounded theory design. It aims to develop a theory grounded in data collected from participants about a process or interactions, right? Example, developing a theory on how healthcare workers adapt to infection control policies. And then the last under the qualitative design is what we call the ethnography design. It involves studying cultural groups in their natural settings to understand their beliefs, behaviors, and interactions. Example, investigating traditional healing practices in a rural Nigerian community, right? Now, let's go to the last arm of the research approaches, which is the mixed method research design, right? Remember, I told you about the explanatory sequential design, which begins with quantitative data collection and analysis, followed by qualitative exploration to explain or elaborate on quantitative findings. Example, surveying patient satisfaction levels and then interviewing some to understand reasons behind their responses. Then we talk about exploratory sequential design. It starts with qualitative data to explore a phenomenon, followed by quantitative testing of patterns or hypotheses that emerge, right? So, example, interviewing herbal practitioners to identify key practices, then using a survey to quantify their prevalence. Of course, the last one under the mixed method design is the convergent parallel design. Here, you collect quantitative and qualitative data simultaneously, analyzes them separately, and then merges the results for comparison or corroboration. Example, combining, focus group discussions and patient satisfaction surveys to assess healthcare qualities. So today, I have talked to you about biomimesis, right? I've talked to you about scientific method. Meanwhile, the opposite of scientific method is what we call scientific consensus, where people they don't go through the processes of scientific method. They just gather and agree on certain things and then push it to the larger community for people to follow. So opposite of scientific method is scientific consensus. Meanwhile, I've talked to you about biomimesis. I've talked to you about scientific methods. I've talked to you about, research approaches, and then we have talked about research designs. So that's what I have to say today. If you have any question, please put it on the comment section and then we trash it. Thank you.